All right. Hi, everyone. My name is Lincon Stamp, and I'm from the Department of Anatomy and Neuroscience at the University of Melbourne in Australia. And I'll be talking to you today about the work we've been doing on developing a stem cell therapy for gut motility disorders, with a particular focus on the pediatric enteric neuropathy Hirschprung disease. So first, just a little bit of background. The enteric nervous system is made up of many different functional types of neurons and glia, and it plays really important roles in the control of gastrointestinal motility, blood flow, and secretion. And as you can see in this whole map preparation of a neonatal mouse intestine, where each of these blue dots represents an enteric neuron, it's actually a really extensive network of neurons, and it's known that there's actually more neurons in your gut than there are in your spinal cord. Developmentally, the enteric nervous system is derived from the neural crest, and in particular from the vagal region of the neural crest. This is from the neural tube adjacent to somites 1 to 7. And during early stages of development, these vagal neural crest cells emigrate from the neural tube and enter the developing foregut. They then migrate within the gut tube, within the gut wall, in a rostrocaudal direction to colonize the entire gastrointestinal tract. And you can see that really beautifully on this right-hand panel, where you can see an embryonic day 10.5 mouse embryo, where all these neural crest cells have been labelled with an antibody against the transcription factor SOX10. And you can see this really beautiful stream of vagal neural crest cells emigrating from the neural tube, entering the foregut, and then migrating within the gut tube to colonize the GI tract. Here we can just see a time course of this colonization in the mouse. So the cells enter the foregut around embryonic day 9.5, and they don't reach the end of the hindgut in the mouse until embryonic day 14.5. So there's two things to note about this. It's actually a really quite a long developmental time course. This is about 25% of the mouse's gestational period. And in humans, it's about 12.5% of the human gestational period. I also note that you'll note that comparing the gut tube at E10.5 to 14.5, you'll note that these cells are migrating along an ever-growing gut tube. So we think they actually migrate the furthest distance of any embryonic cell. And so if you combine this long migratory route as well as this long time course, you have lots of opportunities for things to go wrong. And this is exactly what happens in Hirschprung disease, where these migrating neural crest cells just fail to colonize the distalmost portion of the developing bowel. So Hirschprung disease, or congenital aganglionosis, is characterized by the absence of this enteric nervous system in the distalmost portion of the bowel. So on the left-hand panel, you can see this is an X-ray of an infant with Hirschprung disease following a barium enema. And just to orient you, you can see there the two femurs and the vertebral column, and down the bottom there is the anus. And you can see just above the anus, this constricted region of bowel is the region of the bowel that's missing the enteric nervous system and therefore has no propulsive gut motility patterns. This results in the inability of these kids to pass stool and the accumulation of fecal contents proximal to this constricted region of bowel and the formation of what's known as a megacolon. And as you can see on this right-hand panel, these kids usually present within the first few weeks of life with this massively distended abdomen. So Hirschprung disease affects about 1 in 4,000 to 5,000 live births. It's a multigenic disorder, and interestingly, it displays both variable severity. So some kids can have a very short length of aganglionosis or absence of neurons in their bowel, and some kids can have total intestinal aganglionosis, so no neurons throughout their entire GI tract. And for these kids, life is very, very tough. They're on total parenteral nutrition for the rest of their life. And really interestingly, also Hirschprung displays variable penetrance, so much so that monozygotic twins can be discordant. Current treatment for Hirschprung disease is the surgical resection of this defective aganglionic portion of the bowel, and then reanastomosis or suturing of the distalmost normoganglionated part of the bowel to the anus. And while this surgery is life-saving for these kids, a very large proportion of them have ongoing complications for the rest of their life, including fecal incontinence and constipation. And this presents just not a large financial burden on the families and on the health system, but also a huge psychosocial burden on these kids as they grow up. And so there's been quite a bit of interest in the last few years in the potential of an alternative therapy, and that being cell therapy to treat Hirschprung disease and other enteric neuropathies. And so the idea would be to produce an enteric neuroprogenitor cell, for example, from pluripotent stem cell populations, Or our group and others around the world have shown that in both rodents and in humans, there exists a resident enteric neuroprogenitor cell within the gastrointestinal tract. And these exist out into adulthood and even in the normal ganglionated regions of Hirschsprung's patients. So the idea would be to then isolate these cells, enrich for the appropriate stem or progenitor population, expand them in culture, and then transplant them back into this aganglionic defective portion of the bowel to effectively create a new enteric nervous system where one was congenitally absent. And the ultimate goal, of course, would be to avoid this really invasive resective surgery that these kids have to undergo so early in life. But we think maybe some low-hanging fruit of cell therapy in this context might be using these cell therapy as an adjunct to the current resection, and we may be able to help avoid some of these post-operative complications that these kids have to suffer. And so in our lab, we've been using animal models to build a bit of a roadmap to cell therapy for Hirschsprung disease, and we've been using a number of different animal models to touch on some of these really important points shown here on this slide. But today, I'll be focused on talking to you about our work using a mouse model to look at the transplantation of enteric neuroprogenitors and looking at their migration, differentiation, and functional innervation of the bowel. So where do we get our stem cells from? So we isolate our enteric neuroprogenitor populations from mice where all the neural crest-derived cells are expressing fluorescent proteins. And we isolate these from both embryonic, so E14.5, and post-natal, P4 postnatal gut. We then dissociate the gut, isolate the fluorescent progenitor cells using flow cytometry, and plate them into low attachment plates where they form these beautiful green neurospheres you can see on the right. I have to note these are not neurospheres in the classic sense like a central nervous system neurosphere. They're not clonally derived, so they will contain a mixed population of stem cells, progenitor cells, and also probably some differentiated cells. We then transplant these enteric neurospheres into 2 to 3 week old recipient wild-type mice. And then we leave these mice for up to 16 weeks before we come back and examine what the stem cells have been able to do. So now I'll go through some of our previously published data. We were able to show that these cells were undergoing proliferation following transplantation. So to do this, we transplanted our neurospheres into the recipient animals, and then for 5 consecutive days, injected them with a thymidine analog, EDU. And as you can see, a large number of our graft-derived cells had taken up EDU, and then a population of these had also subsequently gone on to differentiate into Hu-positive neurons, suggesting that they're getting both proliferation and neurogenesis, so generation of newborn neurons from our transplanted progenitor population. Something that's very characteristic of the enteric neuroprogenitor population is that they're really highly migratory, and so we wanted to examine the migration pattern of these cells after transplantation. And we were able to show that our graft-derived cells migrated in a time-dependent manner up to about 6 to 8 weeks after transplantation. So from a neurosphere of no greater than 250 microns in diameter, these cells migrated to occupy greater than 10 square millimeters of the mouse intestine, and their neurites projected well beyond that to occupy about 20 square millimeters of the mouse gut. Again, something that's not exclusive to, but is characteristic of the enteric nervous system is the neurons and glia don't live in isolation. They cluster together in these ganglia. And we found that as our cells migrated away from the graft site, they coalesced to form these ganglion-like clusters, which were composed of Hu-positive neurons and S100 positive glia. Like in the central nervous system, there's a huge range and variety of different neuronal subtypes that make up the enteric nervous system. But the two main subtypes of the ENS are your nitrogen and your cholinergic neurons, which mediate the relaxation and the contraction of the gut musculature, respectively. And we were able to show that subpopulations of our graft-derived cells had generated these nitrogen and cholinergic neurons following transplantation. So this is very exciting. We're getting migration, proliferation, and differentiation, but we really want to know, are the neurons that we're generating from our stem cells following transplantation, are they functional? And to do this, Jamie Fung, our collaborator at the University of Melbourne, performed intracellular recordings from the graft-derived neurons. So while Jamie was recording from these cells, she was able to fill them with biocytin. And so you can see in this left-hand panel, biocytin-filled graft-derived neuron, showing predominantly they showed uniaxonal morphology, and they showed both lamellar and filamentous dendrites, which is just indicative of these cells being a more immature neuronal phenotype. But really excitingly, JAMA was able to show that in response to a step current in this middle panel, the graft-derived neurons could fire action potentials. This particular neurons fired a single action potential, but some of the neurons could fire trains of up to 6 or 7 action potentials. And on the right-hand side of the panel, you can see that they also received fast excitatory postsynaptic potentials, indicating they are able to receive synaptic inputs. So this is really, really exciting. We're generating functional neurons in vivo. But for a cell therapy for a gut motility disorder, you don't need neurons just to be firing action potentials and receiving inputs. You need them to actually functionally innervate the gut smooth muscle, because this is where they'll control the relaxation and contraction that drives propulsive gut motility patterns. And to ask whether our graft-derived stem cells are generating neurons which functionally innervate the smooth muscle of the bowel, we combined optogenetic techniques and intracellular recordings again. So our optogenetic technique, our optogenetic tool in particular, is channel rhodopsin, which is the light-gated ion channel. And so we isolated, in these experiments, we isolated our enteric neuroprogenitor cells, again from embryonic and postnatal intestine of mice, where they expressed both YFP as well as this light-gated ion channel, channel rhodopsin. We dissociated the gut, isolated these fluorescent cells by flow cytometry, and generated these channel rhodopsin expressing enteric neurospheres. Again, we transplanted these into 2 to 3 week old recipient wild-type mice and left them for 4 to 5 weeks. So after this 4 to 5 week period, Rachel Gwynn, who was working in our laboratory at the time, isolated this gut tube and opened it out into a flat sheet. She then took intracellular recordings, this time not from our graft-derived cells, but from the smooth muscle of the gut. And then she looked to see if she could detect any responses in these muscle cells to both electrical stimulation, which will serve as a control, because it'll stimulate all the endogenous neurons of the bowel, and also to see if we could detect any responses in the muscle cells to specific light stimulation of our graft-derived neurons. So before I talk to you about the data, I'll give you a little bit of a background about the neuronal circuitry underlying propulsive gut motility. So the first thing to note on this little schematic is that all of these neurons actually reside within the gut wall. So the enteric nervous system is made up of sensory neurons as well as interneuron populations, as well as these ascending excitatory motor neurons, which mediate the contraction of the gut muscle oral to a food bolus, like you can see here. And this is predominantly through the release of acetylcholine. And on the right-hand side, you can see there are also these descending inhibitory motor neurons, which mediate the relaxation of the gut muscle anal to a food bolus. And this is predominantly through the release of nitric oxide and ATP. And it's this coordinated contraction and relaxation of the muscle which drives propulsive gut motility patterns, including peristalsis. And so just a little bit more information. So the electrical event within a muscle cell underlying a relaxation is known as an inhibitory junction potential, or an IJP, and this is a hyperpolarization of the membrane potential of that muscle cell. Conversely, the electrical signature within a muscle cell of a contraction is the depolarization of the membrane potential, called an excitatory junction potential, or an EJP. And you can see what both of these EJPs and IJP traces will look like in the upcoming experiments. So first, Rachel looked to see whether we were generating any inhibitory junction potentials or relaxations from our graft-derived neurons. So again, you can see here, this is electrical stimulation, which I'll remind you serves as a control, because this will stimulate all of the endogenous neurons in the mouse intestine. And not surprisingly, electrical stimulation resulted in one of these inhibitory junction potentials, which was reduced by the addition of this P2Y1 receptor antagonist, MRS2179, which blocks the ATP-mediated part of the relaxation, and was then completely abolished by the further addition of NOLA, which blocks the nitric oxide-mediated part of this relaxation. And really, really excitingly for us, we were able to show that light stimulation, I'll remind you, so this is selective activation of our graft-derived neurons, also resulted in these inhibitory junction potentials, which had identical pharmacologies to these endogenous inhibitory motor neurons. And here you can just see some of this data quantified here. This is for both the fetal neurosphere recipients and the postnatal neurosphere recipients. And while two things to take away were, interestingly, the light-evoked inhibitory junction potentials appeared to be larger in recipients containing postnatal neurospheres compared to their fetal neurosphere recipients. Regardless, these results show that graft-derived cells contribute to circuits which generate IJPs, or gut relaxations. Of course, next we wanted to see whether we were also generating neurons which contributed to excitatory inputs within these circuits. And excitingly, again, Rachel was able to show, like with electrical stimulation, specific light stimulation of our graft-derived neurons generated these excitatory junction potentials, which were blocked by the addition of this muscarinic receptor antagonist, hyoscine. And these data show that these graft-derived cells, or these graft-derived neurons, contribute to circuits that generate EJPs, or gut contractions. So next we wanted to ask the question, what types of neurons are we generating within these circuits? Are we making the interneurons? Are we making the motor neurons? Or are we making both? And to ask this question, we decided to target the synapse between these interneurons and these inhibitory motor neurons. So it's known that in the mouse distal colon, these interneurons release acetylcholine, which act on the nicotinic acetylcholine receptors on these inhibitory motor neurons, and that this synapse can be blocked by the addition of a drug called hexamethonium. So when Rachel performed these experiments, she was able to show that addition of hexamethonium resulted in a significant reduction in the amplitude of these inhibitory junction potentials following both electrical and light stimulation. And we think this indicates that there's a reduction in the amplitude of the IJP by blocking the synapse. It indicates that they're actually generating these graft-derived channel rhodopsin expressing interneurons in these circuits. However, the fact that this response wasn't completely abolished also indicates that we're generating some of these graft-derived inhibitory motor neurons within these circuits. So this is really exciting. We're generating all the appropriate cell types within these circuits, brand new enteric neural circuits. So this is the data that we saw when we looked at the fetal neurosphere recipients. We were equal measures shocked and disturbed when we saw that addition of hexamethonium in the postnatal neurosphere recipients had no effect, suggesting that postnatal neurospheres do not generate cholinergic interneurons. Our collaborator at the University of Melbourne, Joel Bornstein, then reminded us that there is a population of purinergic interneurons in the mouse distal colon, and that these release ATP, which act on P2X receptors on these same inhibitory motor neurons in the distal colon. So when we blocked the P2X2, P2X3 receptors with a drug called TMP-ATP, we saw a significant reduction in the IJP amplitude, indicating that while these postnatal neurospheres don't generate these cholinergic interneurons, they do generate purinergic interneurons. And here we just have the summary of these electrophysiological experiments. And the takeaway is a very busy slide, but the takeaway is that neurons derived from progenitors isolated from the embryonic gut, their motor neurons and interneurons have identical pharmacologies to the endogenous neurons of the mouse distal colon. Neurons derived from progenitors isolated from the postnatal gut, their motor neurons have identical pharmacologies, however, they look to have limited interneuron populations. So in conclusion, hopefully I've been able to show you that the enteric neural progenitors transplanted into the colon of recipient mice have generated functional excitatory and inhibitory motor neurons whose pharmacologies match those of native motor neurons. Interneurons were also generated, however, the age of the donor influences the neurotransmitter phenotype. But we think the generation of functional motor neurons and interneurons from progenitors isolated from the postnatal bowel is clinically really important because it suggests that patient-derived cells might be a possible source to treat Hirschprung disease and other enteric neuropathies, such as achalasia and some forms of diabetic gastroparesis. And future studies are involving transferring all of this experiments into a Hirschprung model recipient mouse, and also expanding into larger animal models, and also looking at methods to analyze actual propagating motility patterns in these recipient mice, rather than the intracellular recording electrical signatures of relaxations and contractions. And so I'd just like to thank a few people, my boss and mentor, Heather Young, Marlene Howe, my co-lab ed, Rachel Gwynn and Jamie Fung, who performed the electrophysiological studies, which I really think sets our work apart from our contemporaries. Rio Hotter, who helped get this research off the ground in the very beginning. Professor John Finesse from our department, who helped develop the surgical techniques for transplantation. And our other collaborators at the university, and in Hideki Inamoto from Japan, who helped to generate some of the mice used in the study. Thank you very much.